Welcome to this year's last minute crash review for AP Biology. In this video, I have updated tips and pointers for the AP Biology exam. I'm going to go over some really commonly questioned content that shows up on the test and review some basic skills that might pop up on the test this year. Remember, this is a fast review and it's definitely not meant as introductory content, so if you need to go back and do a deeper dive into any of the topics listed in this video, make sure to dig into some of my other resources or the resources provided by your teacher. And remember, AP Biology is a trademark owned by the College Board, which is not affiliated with and does not endorse this video. I have lots of other AP Biology resources and test review materials, a lot of which I've updated this year, so please be sure to check those out on my channel, especially if you have some extra time. I do talk fast. If you're watching this video, that is just a warning. You may want to slow it down, or if you're pressed for time and you're watching on 2x speed, just be aware that it will seem really fast as I go through. Remember, this video is going to go over a lot of content, but that is not the only thing that is tested on the AP Bio exam, especially in the FRQs. You're going to have to use some of these scientific practices throughout the free response questions. There are 2 long and 4 short AP Biology FRQs currently, and these are the skills that are going to show up on each of those. As far as topics go, on the AP Biology exam, you'll see all of the topics from all 8 units in AP Biology with similar weights on the exam, but remember that natural selection does show up a little bit more than the others, so be aware of that and make sure you devote time to review for that topic. Let's cover some content or themes that frequently come up, and these are going to come up in no particular order, so be aware that this is not the order at which you might have learned them in your class. Let's start with the central dogma of molecular biology. Hopefully you remember a little bit about protein synthesis from your original biology class, but in AP Bio it gets a little bit more complicated, where the enzyme RNA polymerase is going to synthesize mRNA molecules in the 5' to 3' direction by reading the template DNA strand in the 3' to 5' direction. So we have our RNA polymerase synthesizing that molecule, the mRNA leaving the nucleus, and then undergoing certain modifications, which we'll get into a little bit later. And then in the cytosol at the ribosome, we have our tRNA bringing around our amino acids, and it recognizes each codon with an anticodon on the tRNA, and that amino acid chain is built from the mRNA transcript. In prokaryotic organisms, this happens all at once. Transcription is coupled to translation, but in eukaryotic organisms, we have this occurring in our different locations. Once we have our amino acid sequence, we can undergo even more protein modifications to get the final functional protein. Now, again, there's lots of regulation of genes and transcription and translation. Some types of gene regulation in prokaryotic organisms include operons. Examples you might have seen in your class are the trip operon and the lac operon. The trip operon is an example of a repressible operon because its transcription is usually turned on, but it can be repressed when specific molecules bind allosterically to the regulator proteins. Inducible operons, like the lac operons, are usually turned off, but they can be stimulated or induced. So in our image here, we see a simplified version of the lac operon. Here, transcription is blocked and the gene is turned off, meaning it's not able to be transcribed. So there's no translation either and no functional proteins, these enzymes here. But if an inducer is present, in this case allolactose, it's going to bind to the repressor. The repressor will be removed, and it allows RNA polymerase to continue to transcribe these lac genes. Now, there are many levels of gene expression and regulation in eukaryotic organisms, and there's many points at which the transcripts can be changed or modified along the way to becoming active proteins. There's even pre-transcriptional regulation that can occur. A big one to know is part of RNA processing, where we have introns and exons. Our non-coding sequences are introns, and then those are later removed so that we only have exons in our final mRNA transcript. With alternative splicing, we can get multiple versions of mRNA from the same original gene. Jumping to a later topic, biotechnology, be sure you know the basics of what PCR is, gel electrophoresis is, DNA sequencing, and then bacterial transformation. You won't have to detail step by step how these occur or the laboratory procedures for these, but you should know the purpose. For example, PCR is the amplification of DNA to make lots and lots of copies from a small bit. Gel electrophoresis is the separation of our DNA molecules based on size so that we can see any patterns after they're cut with restriction enzymes. This is useful for evolutionary analysis, as a step in genetic transformation, or for forensics and crime scene analysis. Bacterial transformation or genetic transformation involves the removal of a gene of interest from a particular organism, inserting it into a plasmid so that our gene of interest can then be taken up by our organism that will later go and express that gene as a protein. Make sure you know the 4 main categories of biological macromolecules: nucleic acids, proteins, carbohydrates, and lipids. Know their monomers, the structure of their monomers. So nucleic acids are made of nucleotides. Proteins are made of amino acids, carbohydrates are made of monosaccharides, and lipids are made of fatty acids and glycerols. Organisms use feedback loops to maintain their internal environments and respond to internal and external changes. Feedback loops help organisms regulate homeostasis, and the failure to maintain homeostasis can cause disease or death. Negative feedback loops are very common in organisms. These are going to result in the slowing of a process or getting something back to a set point. So for example, blood pressure, blood sugar regulation, temperature regulation, so that the body can return to the levels at which it needs. Positive feedback loops are an amplification of a particular output or signal. It's going away from homeostasis. So for example, increased contractions during childbirth, the production of milk in mammals, or even the ripening of fruit. Cellular energetics is another really important topic in AP biology, and beginning with cellular respiration and the steps involved. The three main parts are glycolysis, the citric acid cycle, and then the electron transport chain with oxidative phosphorylation. Glycolysis occurs in the cytosol outside of the mitochondria. Our citric acid cycle occurs in our mitochondrial matrix, and the electron transport chain occurs across the inner mitochondrial membranes in order to establish a proton gradient for ATP synthesis. Remember that oxygen is our final electron acceptor at the end of this process. Photosynthesis and cellular respiration are highly connected, especially within the carbon cycling of the biosphere. Cellular respiration metabolizes organic molecules, which are sugars, and returns carbon dioxide to the atmosphere. Carbon dioxide is our byproduct. Photosynthesis uses carbon dioxide, so it removes CO2 from the atmosphere and fixes carbon into our organic molecules or sugars. Remember, photosynthesis is the process that converts solar energy into chemical energy. Light energy is converted from photons into the energy that we need, and carbon is fixed into organic compounds that the cell can later use in cellular respiration. Remember, plants perform cellular respiration as well as photosynthesis. You definitely should know the overall equation for photosynthesis, and be sure to review the steps of photosynthesis to a certain degree, including both the light-dependent and the light-independent reactions. So knowing where the inputs are used, or the reactants that you saw in the previous slide, knowing where those are used and knowing where the outputs are generated is important. So for example, for water's role in photosynthesis comes up in the light-dependent reactions, the first part there, within the thylakoid, and when the electrons are released from water molecules, they're transferred to photosystem 2. Evolution is a main theme that pops up again and again on the AP Bio test. You should probably study endosymbiosis. Remember, this is the idea that eukaryotic organisms arrive due to engulfing smaller prokaryotic organisms and forming a symbiotic relationship with them. There's evidence for this. Some of that is that we have double-membraned organelles, mitochondria have their own DNA, the fact that almost all existing organisms perform glycolysis, and that it's an anaerobic process means that it also could have occurred when there was no oxygen in the early days of Earth, and it's one of the most universal processes of respiration. Some common topics and themes that show up related to molecules and molecular biology. Enzymes at high temperatures will unfold or denature or lose their shape and lose then their function. So remember, an increase in temperature above the optimum range for the enzyme can definitely alter the shape of the active site and reduce a reaction rate. Water has a lot of very special properties, such as high heat of vaporization, the fact that it's polar, surface tension, just to name a few. This can relate back to a lot of reactions and processes within our bodies and cells, including homeostasis. For example, that high heat of vaporization is involved with sweat because the water in sweat absorbs a large quantity of heat during that liquid to gas phase change, which is going to lower body temperature and bring us back to the temperature that our body needs to be. You definitely want to be able to recognize how different molecules are going to be able to cross the membrane. For example, small nonpolar molecules like carbon dioxide and oxygen can freely pass through membranes. So speaking of membranes, cells have membranes that allow them to establish and maintain internal environments that are going to be different from their external environments. And then we have internal membranes that are going to help with different cellular processes, and it minimizes different interactions that are going to be competing within the cell and increases our surface areas where our reactions can occur. And we can also use this as evidence for the endosymbiosis hypothesis. But all of these themes are important when we talk about organelles. Now, these are the organelles that you really should be able to know, recognize, and understand how their functions work for AP biology. There's a lot of other organelles out there and some major ones that are not on this list list, but not on the exam. These are the ones that might show up and you might be tested on. Otherwise, if they're not on this list, you can forget about them. Out of this list, the most important ones you really need to know and recognize are cell membranes, mitochondria, and chloroplasts. When we're talking about cell membranes, remember these are very complex. They go beyond just a phospholipid bilayer. They have integral proteins that are embedded in the membrane for transport, cell surface reception, cell identification, other activities. They also have carbohydrates and cholesterols in the membrane, so be able to recognize how membranes are going to contribute to cell structure, function, communication, a lot of other themes and processes. Mitochondria and chloroplasts, of course, are going to show up when we talk about cellular energetics. We already talked about mitochondria and chloroplasts a little bit with endosymbiosis, but make sure you're very familiar with them, their role and their structure for both cellular respiration and photosynthesis. All right, going back to cell membranes a little bit more, be able to recognize the phospholipid structure, how we have a polar group of the molecule, it's here in red, and then our little squiggly hydrophobic tails that are going to form the internal part or the middle of our phospholipid bilayer. A lot of students really get worried about cell signaling pathways. These are not that complicated, but do remember cell signaling pathways are characterized by a signal, a transduction, and a response. That signal could be something generated by a unicellular organism or could be part of multicellular signaling pathways. Signaling pathways are highly specific and regulated, and one single signaling molecule can cause a cascade effect, releasing thousands of molecules in a cell. And these pathways we know probably evolved from a common ancestor millions of years ago. We can even look at similar molecules in pathways in different species and use this as evidence to investigate different species and how they might have evolved from a common ancestor that used very similar signaling molecules. Now, the result of a signal transduction pathway could be something like gene expression or the production of a hormone or suppression of a genetic activity or even apoptosis, which is programmed cell death. Jumping back to more molecular structure, DNA, make sure you're very familiar with the structure of the nucleotide itself, and then of course the full DNA molecule. In the DNA molecule, we have a sugar phosphate backbone, and our bases are on the inside, connected by hydrogen bonds. Cytosine is always going to pair with guanine, and adenine is going to always pair with thymine in DNA. DNA is structured as an anti-parallel double helix going from opposite 5' to 3' orientations. And to contrast it, RNA is single-sided. It does have structural similarities to DNA. It still has a sugar, a phosphate group, our nitrogenous base, but RNA has uracil, DNA has thymine, DNA obviously is double-stranded, and DNA usually is just staying put in the nucleus in eukaryotic organisms, whereas RNA is going to leave the nucleus to perform the other steps of protein synthesis. Another hot topic is the cell cycle and checkpoints. There's a number of internal controls or checkpoints that are going to regulate progression through the cell cycle. You don't need to know specific cyclin-dependent kinase pairs or specific growth factors, but do know that if something is wrong or if there is an error detected within the cell cycle, typically the cell will commit apoptosis, again, programmed cell death, but errors in that cycle or errors in recognizing those steps could also lead to uncontrolled cell growth, disease like cancer, or we have uncontrolled cell division or unregulated cell division. Now, speaking of cell division and reproduction, mitosis is going to produce two genetically identical daughter cells. Make sure you review different types of reproduction. Meiosis is going to produce cells with half the normal amount of genetic material, generally going from diploid to haploid cells, and these are gametes or sex cells. There's a lot of different steps that contribute to genetic diversity. First of all, we have independent assortment, where we have a lot of different possibilities of orientations of how the chromosomes could line up. There's random fertilization, which again happens after, but we need to be able to create these haploids to be fertilized beforehand. And then during crossing over, we can have a bunch of different combinations of information. Familiarize yourself with errors or consequences if some steps in the process of meiosis go wrong. Make sure to review cell transport situations. Well, water move by osmosis from areas of high water potential to areas of low water potential. This is probably going to be recognizing where water would move within a certain situation is probably more likely to show up on the exam than an actual water potential problem. As far as genetics goes, inheritance patterns that might show up include single gene or monohybrid inheritance. Dihybrid inheritance is a possibility as well, where we're tracking two genes. sex-linked inheritance, where we have a trait. Generally, it's going to be an X-linked, sex-linked trait that we're tracking. And remember, males only need one copy of those in order to display the trait. Females need two if it's recessive. We can talk about genetically linked inheritance. These are when two genes are close together on chromosomes, so typically they're inherited together. And then inheritance of things like traits in mitochondrial DNA. Remember, your mitochondria is going to come from the egg or the ovum, and so that is going to be inherited from the mother. As far as the ecology and the environment, there's a lot of related topics that could show up on the exams. A very common one is environmental impacts on molecular functions or environmental impacts on gene expression. So recognize how certain genes can be turned on or off or have extra expression due to certain environmental factors. Environmental pressures can also strongly influence natural selection, so make sure you connect those ideas. And then, of course, population interactions like symbiotic relationships, predation, and lots of community ecology topics could show up here and there. So make sure you know how organisms can interact with each other and some of the typical symbiotic relationships that you might see, such as mutualism, parasitism, and commensalism. All right, we're getting into some general tips, and so these are things that I'm just going to go through very quickly to make sure you remember for the exam. Remember to write in complete sentences. You do have to write in sentence form, but you don't need to write full paragraphs. Make sure you avoid bullet points, lists, outlines, drawings. Don't waste too much time obsessing over perfect grammar and spelling, and points are not counted off for spelling errors as long as the graders can understand you. Don't waste your time restating the questions. Remember, the two long FRQs do have more points, so you want to try to answer those to the best of your ability. You do want to try to answer all of the FRQs, but the longer ones, there's more points opportunities on them. Make sure to review experimental design because you will be asked about that on at least one of the FRQs, and make sure to avoid lengthy explanations. Take out words like thus, just write in simple sentences that are straightforward and to the point. Avoid words like love, prove, because that's not a great word statistically to include, and any absolutes like always or never, and make sure to gut check your response just to see if it makes sense before you submit your answers. In experimental design, make sure you recognize how we measure dependent variables. Remember, these are the things that are measured and how we take our data in the experiment. Independent variables are the things that are changed, tested, or manipulated. Think I, the experimenter, what am I changing for the independent variable? And D, what am I measuring for my data for the dependent variable? When it's asking you to make a prediction, make sure you state something specific. Don't just say, I think this variable will change, or I think there will be a difference. You do want to make sure that you include whether it's an increase, a decrease, death, growth, whatever. But you don't need to include your justification unless it asks you to justify, which it probably will on a particular step of the FRQ. But answer what the question is asking. Don't do more, don't do less. Now, you will have to create some sort of graph on the AP Biology exam. Make sure you choose the most appropriate graph to explain your data. I do have a whole video on that if you'd like to check it out. Line graphs usually show a change over time. Bar graphs usually display the comparison of different categories. You might have to draw a scatter plot as well. It is unlikely you would have to create a pie chart for the AP Bio exam. More than likely, you'll probably have to create a line or a bar graph. Just make sure you choose the appropriate one for the exam. Be sure you're familiar with your place values. This is just a slide to remember which ones are which, because sometimes if you're calculating something, you may be asked to round to a specific place value. For example, round your answer to the nearest hundredth place. You will not be asked to use significant figures, so don't worry about that on the exam. Make sure to review error bars and know how to determine if there's an overlap between the bars of two or more sample means. Remember, if error bars are not overlapping, there is a statistically significant difference between the values, which means our experimenters can consider the measured values as not the same. But if the error bars are overlapping at any point, then we can probably say there is not a statistically significant difference between the values and probably not a statistically significant difference in the data. So if we look at this graph here, rarely or never, and then more than weakly, these error bars do not overlap, but most of the other error bars do at some point. As far as the math, there are some equations on your formula sheet that you probably shouldn't waste too much time on because it's not likely they'll show up on the exam, and that's standard deviation, standard error of the mean, surface area equations, and pH equations. They are on the formula sheet, but it's not likely you'll see them on the test. Equations you should be familiar with are things like the chi-square, the Hardy-Weinberg equations, water potential, population growth, and carrying capacity, and maybe Simpson's diversity index, though it's probably not going to be on your test. I do have a longer video going through examples of each of these, so be sure to check that out if you're interested in doing a fast AP Biology math review. But really quickly, I wanted to highlight chi-square because this is something that a lot of students get stressed out about. It is used in science to test if data that you observe from an experiment is the same as the data that you would expect to come or predict from the experiment. What's really important in chi-square is to know how to do it and how to interpret the results. Sometimes you'll use this in a genetics problem, and if you're using it in a genetics problem, you'll probably state a null hypothesis, something like the data are consistent with the predicted method of inheritance. You calculate your chi-square value, and then you make sure you know your degrees of freedom, which is the number of categories total minus 1. In this particular case, if we're looking at colors, we have 1, 2, 3, 4, 5, 6 colors minus 1. That would be 5 degrees of freedom. After we got our chi-square value, we would look at that on our critical value table, and we would see if our chi-square value was greater than the critical value in the chart. Please go back and check out my chi-square video or my AP Biology math video if you want to see a full example of this. Here are some examples of null hypotheses which you might need to be able to state. Again, if you're talking about a null hypothesis in genetics, you would say something like the data are consistent with the predicted method of inheritance. If you reject a null hypothesis, that means that there is some difference in the data in the experiment and that the variables did have an effect on the particular outcome of the experiment. Now, we've just hit on a lot of content very quickly, so be sure to check out some of my other resources for a deeper dive into test-taking skills and the content that you might see on the exam. Make sure to leave a comment below if this video has been helpful. Good luck on your exam, and I'll see you on the other side.